Okay, so it's a great pleasure to be here. So I'm going to tell you about some work done in collaboration with Simona Coco and Lorenzo Posani, who is finishing his PhD at ENS, with Karel Jezek, who is running an experimental lab in Pilsen, in the Czech Republic, and Sophie Rose, who was a PhD student and is now at CISA in Trieste. So, but before I start to tell you something about what we have done on continuous attractors on neural networks, I'd like just to say a few words, because obviously this is not any kind of conference. This is a conference in the honor of Giorgio. And obviously all my feelings and recordings about Giorgio are related to my time here as a postdoc, a long time ago now. And many things I'm remembering, and there are two of them I would like to tell you about. First of all, that was a really great time for me, I mean socially and intellectually. It was a time, 94, 95, more than 23 years ago, where many people I met here in Rome have become colleagues and friends. So some of them are here, Laetitia is here, Roger is here, Andrea I met before in the corridor, and many other people are not here, but are still very important to me. I mean, Nicola and Juan Jesus and David Lancaster and many other ones. And I think I'm very grateful to Giorgio, to Enzo, and to Miguel for making this possible, because I think that was very important and very stimulating. The second thing, and I thought about that when I was preparing this slide, that the scientific discussion with Giorgio was very important. So it turns out that I do remember really well my first scientific discussion with Giorgio. Probably you don't remember, and in fact, that was not interesting to you, probably, but it was for me. So what was the first discussion? So let me see. Okay, so what was the first discussion? So it turns, that was connected to some work I was doing at the end of the PhD with Marc in Paris. So Marc had told me that I should be studying the random field Ising model in the high temperature phase. So I don't want to say too much about that, but just in a few words. This is a model which is very complicated, which has been studied over decades, which is not totally clear. And in particular, in a high temperature phase, you would expect the system to be in a parametric phase, and the two correlators here, the two propagators should have the same correlation length, the diagonal and the off-diagonal ones. So here the brackets means the thermal average, and the overbar means the average of a disorder, as usual. And at some point, the two correlation lengths will start to be different, and that's a signal of the onset of a glassy phase. That was what the feeling at that time. And so I used a paper, based on a paper by Marc and Peter Young, in order to compute these correlators, I used this self-consistent screening approximation of Bray and Moore, which is some kind of mapping of the Ising model to O of n spin. And when n, which contains all the resummation of all diagram to order 1 over n, and sum to all powers, but not all of them. And you can solve the self-consistent equation for these propagators, find the correlation lengths, and this is what you get as a function of a size L of a lattice. So that was a size you see running from 2 to 20 to the cube. And you know, after 6 months of work, you get this, and you ask whether these two numbers become equal when L goes to infinity or not, which is a bit depressing, right? So as a first discussion, I went to Giorgio and I told him about that, and immediately he told me that, but you should not plot the results this way. Obviously, this is not the right way to plot the results. You know that there will be finite size effects which will scale like exponential minus L of xi. So just replot this as a function of exponential minus L of xi instead of L. So at that time, there was no graphical computer, so I had to program and go to the basement, to the printer to see the curve, and the result is this one. When you do that, and you see that then if you do that, the two masses go exactly to the same point here, which means that they are really equal, and that you can check with a lot of digits. So why I am telling you this is a trivial example, but I think it's just is a good summary of the interaction with Giorgio. So many times during the postdoc, when I got into his office, I was on the left side of the slide. Some kind of a little bit depressing and confusing situation, and Giorgio had the right insight, either technical, conceptual, the right reference, on the right question to ask next, what to be studied, and so on, to move to the right side. So it was great, so I'm very grateful to you for that. Okay, so now going back to the topics of the talk, I'm going to tell you about neural networks, which I think were a very important application of spin glass techniques in the 80s. had a little bit decayed over the years and now are coming back full steam. And there are two reasons for that. One is the success of machine learning, so there is a need to understand a lot of things, and especially in deep neural networks, but in many kind of architectures. And the other one is that the techniques in experimental neurobiology have nothing to do now with what they were in the 80s. So many of the questions which was purely speculative in the 80s now can be addressed experimentally, and this is what I'm going to tell you about, just to show a few things. So what is the question people ask in the 80s? Is suppose you have a neural network like that, so with a set of connection between n neurons. So for simplicity, we can assume the neurons are 0, 1 variables. So the neural space is the n-dimensional hypercube, and one point here is a corner of this hypercube. So it's a string of n bits. And so in the Hopfield model, and in any case, you don't care too much about dynamics. You would like to be sure that the dynamics is actually leading you to a point of interest. So for instance, one configuration of the activity you would like to memorize. Okay? So the question was how to engineer the connection between the neurons in such a way that you can actually be sure that the dynamics will bring you to the points here of interest, the memories inside the network. So the spin glass techniques were applied just a few years after this Hopfield introduced this model, and that was, I think, one of the very nice application of spin glass techniques to biology or to theoretical biology. But of course, there might be attractors which are more complicated than fixed points. There might be continuous attractors, or let's say finite dimensional attractors. So you could imagine that your neural space here, that is the set of all possible neural configuration, has now not fixed point, but has a fixed manifold, let's say fixed manifolds, which are attractors of the dynamics. And depending on the initial condition, you can land here on one point along this manifold or another one. Okay? So this model was first proposed in the case of a single continuous attractor by Amari in the 70s. And then it was proposed in the case, in the application to representation of space in the brain by Tsodix and Sosnowski, and it was also solved in order to compute how many kind of attractors like that you can store in single neural networks by Battaglia and Travis at the end of the 90s. So one question I'd like to address, and we have been addressing in the recent years, in particular with Sophie, Jose here, is what happens when you have transition between these continuous attractors. So these transitions can be spontaneous transitions, or they can be driven by some particular inputs or some particular forcing, which will bring you from one attractor to another one. And the questions are, how can you go from one attractor to another one? And if you go back, for instance, where will you land compared to the initial position? So what I would like to convince you is that these questions actually can be asked in connection with real experiments in rats. So now I'm going to the presentation of the system we are studying. Yes?
So the difference at one point on the attractor is one fixed point, or once you are on the attractor, you move on the attractor?
Okay, so in principle you have some kind of zero mode. So it's a fixed point, but it's marginally stable. So you can diffuse. There will be diffusion along the attractor. Is it clear?
If there is some noise.
If there is some noise, yes. And then I go back to the question whether the attractor is perfectly zero mode, very flat, or it could be a little bit bumpy. Okay, so what I'm interested in is something which has been discovered a long time ago in the 70s and by John O'Keefe and collaborators, and he was given the Nobel Prize for that a few years ago. So the hippocampus is a cortical region which is common to all mammals and essentially present in some slightly different configuration in all vertebrates. So just to give you an idea, there are about 300,000 neurons in the rat hippocampus and about 2 orders of magnitude more in human hippocampus. And it was known for a long time that damages in this cortical area impair the capacity to have spatial memories, to acquire new memories. So in fact, what O'Keefe did is he put some electrodes inside the hippocampus of rats, and he could see that there are some specific neurons here in the hippocampus, which are called place cells, which respond to position in space. So in a schematic way, here what you see is that a rat is moving in some arena here, and one particular neuron, which is the blue neuron here, will be active when the rat is inside this particular region, which is called the place field, and it will be silent if the rat is outside. For another neuron, this red neuron, it will be another region here. So it's another place field. So the idea was immediately that all the place cells can actually give information about the position of a rat to the rat, so that is a way to actually establish a map of a space. Okay? But actually the story is a bit more complicated than that, And here I want to show you two more experiments to see how it becomes complicated. One experiment by Smith and Mizumori from 2006, where you see exactly the same kind of experiment as O'Keefe did. So now the space is not a circular arena, it's just a cross. So the rat is starting from one of the extremities of the cross and is moving. So you can do 10 trials when you put the rat somewhere and let it run, okay? And then when it arrives at the end of another arm of the cross, then you stop the experiment and repeat it 10 times. And you do another block of 10 trials, exactly in the same way. And if you record in the hippocampus, for instance, you will find a place cell which is active here in the east arm of the cross. And this is the same activity, the same place field for the second block of 10 trials or for the first block. There is no difference between the two blocks of trials. But now, train the rat to do something more complex, not only wandering in the environment, but actually the task the rat has to be trained is the following. During the first trials, there is a reward in the east arm here, and in the next block of 10 trials, there is a reward in the west arm. So the rat has to learn that it has to go 10 times to the east arm, and then 10 times to the west arm, okay? And now you can record the activity of one place cell in the hippocampus during the two blocks of trials, and you see, for instance, for this cell number D here, then the cell is active actually where the reward is. So its place field is changing there is a remapping of the place field in between the 10th and the 11th trial. Because actually what the task the rat has to do is changing, so the place field is actually moving according or changing according to the change of task. This cell, for instance, is active only during the second block of 10 trials in the middle of the cross, and it's not active in the first block. So what I mean here is that the place cells are indicative of position, but in a given context. If you change the context, then the mapping between position and activity is changing. So now we see that we are actually looking at a map, not only in physical space, but in a more complicated space, where some directions are context, whatever that means. So the next block of experiment I want to show is this one, which I like very much. So it's an experiment by Diba and Buzzaki in 2007. So here you have a rat running along a linear corridor. There is some water here and some water here, so two reward spots. And you see, as the rat is moving here, you have the spikes emitted by the cells. So you have 13 place cells, which have been ranked according to the centers of the place fields. So during the physical motion of a rat, so here you have time in seconds, so it takes about, I don't know, 5 to 6 seconds for the rat to go from one extremity to the next one. Then you see the cells which fire one after the other, okay? But now, if you look at what happens when the rat is actually immobile here at the red spot here, then you see that the whole sequence of place cells is activating also, but on a much shorter time scale. It has nothing to do with the physical motion because the rat is immobile, and the time scale is about a few hundreds of milliseconds, 200 milliseconds, which is much, much faster, at least by 20, factor 20, compared to the physical motion. So what the rat is doing is just planning the forward motion, and you get also some backward replay at the end here. But clearly that means the network of place cell is able to sustain some activity without any physical, without any input coming from the physical motion, okay? So it's not simply that when the rat is somewhere, it receives some input and then it activates the corresponding place cells. Here it doesn't move and the whole network is activating. So that means that through training, some network has been generated in between the place cells, which is able to sustain the activity. And of course, there might be inputs coming from other brain areas. And the question now is, since we have a single hippocampus and we have many maps and many contexts, how are we able to store all these maps in the hippocampus in a single network? So we are back to the question Hopfield was asking a long time ago, but in a slightly different context, because now a map is not a fixed configuration of activity, it's a manifold. So it can be a two-dimensional manifold for rats. It will be a three-dimensional manifold for bats. There are also experiments for bats which show exactly the same thing, place cells in bats. So the experiments we were interested in is the one by Jezek and collaborators in the Mother and Mother group, which was published some years ago, 5 years ago, 7 years ago now. So about 30 neurons were recorded in the hippocampus of a rat. In this situation, so the rat during the training phase is moving in between in two boxes which are connected by a corridor. So typical dimension here is 60 centimeters of this box. And the boxes are exactly identical, except that they differ only by the light conditions. You have dim light on the roof of the box here, and dim light on the sides of the box here. So the rat is trained for a long time like that, and then if you record the activity of one place cell, what you see that place cell number 1, let's say, will be active when the rat is somewhere in the bottom corner of this box here. It will be active at a different position, of course, in this box here. Okay? So experimentalists have to make sure that the corridor is long enough in order that the rat is sure that the two environments are different. Okay? Otherwise, you will have twice the same spot here. So that means that the same cell is coding for two different positions in the two different boxes. So that's a training phase. Well, once you train the rat for weeks like that, then you can have the test phase where there is a single box, and there is a light switch which allows you to drive the rat from the light condition of box A, let's say, to the light condition of box B. Okay, and then you can monitor the activity in the hippocampus and see how the rat is teleported to the other contextual environment. Is it clear for the experiment? Okay, so one thing we wanted to do is to do some decoding. That's the obvious thing to do. Then I can tell you how the people who did the experiment did that. But let's talk about how we did that first. So what you would like to do is at a given time, or let's say at a given interval of time, which is about 100 milliseconds, so there is a good reason to look at this time scale, but I'm coming back to that later. You have a population activity here, which means for each neuron, you have a 1 if it's active or 0 if it's not active during this time interval. And you get this configuration, and you would like to know whether the rat is actually mentally in the representation of box A or in the one of box B, to decode the environment where the rat is mentally. So it turns out that the average activity of the different cells in A or B, they are very similar. Okay? So you cannot do an independent cell decoder. That won't work. But what you can do is the following thing, is that if you look at box A, for instance, in the first environment, if I look at these green and orange neurons, it might be that there are two place fields will overlap. So that means that when the rat is here, they will be both active at the same time. While they are far away in box B, which means they will never be active at the same time. It's very unlikely. So the pattern of correlations between the different cells is actually very specific of one environment. Okay? So we can use this in order to decode, without any knowledge of the position of the rat, in which environment the rat is. So to do that, we use a very simple Ising model. So you have an Ising model. So this is not supposed to mean anything in terms of physiological interaction, just a way to actually give a score to a particular configuration of activity in one of the two boxes. So here you have local fields, you have effective interactions, and we just fit these parameters exactly as Martin shown this morning, except that it is simpler here because we have only Ising variables, not Potts variables, but it's exactly the same idea. And we do that for box A and we do that for box B. So we get two Ising models, which actually doesn't make much sense because there is a single hippocampal network, so in principle we should use a single one. But if we want to do decoding, it's fine. Okay? And now we have the log of this is a likelihood of the activity configuration given the fact that we are in A or B. So for instance, if I plot the difference between the two logs, so log P of A minus P of B, what I will have is something when I am in reference session A, then this is something which, okay, fluctuates over time, it's almost always positive. In B, it's almost always negative. And the histograms are well separated. There is a little bit of overlap here, and of course, this is due also to the fact that we have a limited number of recording neurons. If you have a higher number of neurons, then they will be more and more separated. But still, so it allows you to decode whether the rat is in A or B. So what happens if you use this decoder during the test phase, where you have a light switch from A to B, then this is what you get, for instance. You see, for instance, delta L is strongly negative before the light switch, so the rat is in B. Then you move the light condition to A, and eventually after a few, let's say 10 seconds or whatever, you'll be somewhere here in the A condition, but in between you've seen some flickering, some oscillation. So that means the mental representation is actually confused between A and B. And then it stabilizes in the right representation, which is the one in agreement with the external light condition. Okay. So now one question is, during this instability what about the decoding of position? So the rat is confused about whether it's in A and B, but is it still able to decode or to have a good information about the position? So how to decode the position? So I won't go into details, but you can imagine how this is done. This is easy. In each map, in each environment, you have some kind of quantity, which is the average, for instance, firing rate of neuron I when the rat is in X. This is exactly this rate maps I showed before, okay? So that means that if I know the activity here, I can use a decoder, for instance, independent cell decoder or something very simple, which will tell me what is the probability of this given X, and then I will maximize over X and find the position. But of course, I must know what set of rate maps here to use, what set of place fields. There are two of them, one for A and one for B. So if I use A, I will decode some position. If I use B, I will decode another one. So if I do this decoding, here you see the result. So this is the decoded position X as a function of time, and in the bottom panel you see Y as a function of time. So the real position is the red trajectory, and the blue one is the decoded one. So if you use the place fields which correspond to V1 of a light condition, you see a very good agreement before the light switch, and then when you switch, then it's really bad until you wait long enough, and then it becomes okay again. But in between here, there is a huge error. But now if you use the place fields which correspond to the decoded map, to the map we have inferred with this delta L I showed before, then now the agreement becomes very good. Which means that there is a huge confusion in terms of representing the position according to map A or to map B. But the way the position is encoded is precise at any time, at all times. So you can imagine you are conveying some information, you switch languages at any time, so very quickly, abruptly, but the information is always correct. Okay? So this is what is going on. So in the second part of the talk, I would like to present a model where we could have such kind of phenomenology, because it's not totally obvious how this can arise. So essentially from a theoretical point of view, this is exactly the same slide as before. We have a network, so because we have a single hippocampus, we have two attractors corresponding to the two maps. There are two planes, okay? And the rat is, I don't know, moving, for instance, in context A. If it is at position X, and then the neural configuration will jump to V1 corresponding to the same position X on this attractor B. Then it will go for some time along this attractor. Then when the rat is in X prime, it will go back to attractor A exactly at the right position X prime. Because the knowledge, the information of position is right at any time. Okay? But there are back and forth jumps in between the two attractors. So we have two questions here. First, how to model the storage of multiple continuous attractors in a single network, and then how do we understand the fact that the model jumps at the right neural configuration coding for the right position? It could jump anywhere. Okay? So how can we make this precise? Okay, so about the model. So the model was, that's a model we introduced, but actually it's some kind of version of model which was introduced before, especially by McNaughton in 97 and Battaglia Travers in 98. So just to make it more spin glass-like, and so it's very similar to the Hopfield model, except that in the Hopfield model you want to store fixed point configuration, which means that the objects you are interested in are this pattern, psi mu. So I is the index of a neuron, mu is the index of a pattern, and the Jij will be, the couplings will be a sum of a mu, of psi mu, psi j mu. So this I can write it as a Jij mu. It's an elementary interaction matrix for the pattern number mu, and it's a rank 1 matrix. Okay? Now here, the difference is that we do exactly the same additive rule, except that the elementary matrix for each environment are not rank 1, but they are matrix which code for a continuous attractor. So they are similar to the matrix, to the idea which was introduced by Amari a long time ago. So we have matrices actually here, which depends for each neuron I and J. We have a function of the distance between the centers of the place fields of the corresponding neurons I and J in the environment. Okay, so each neuron I and J will have a place field in environment 1, in Xi and Xj, and we have an interaction which decays with the distance. So why do we do that? Because actually it's an idea that people had a long time ago in physics. If you think, for instance, of liquid-vapor transition, the work on liquid-vapor transition by Lebowitz and Penrose, they introduce models of lattice gases, which are exactly the same one. You have particles on a lattice, and you put an interaction, Jij, which is a function of the distance between the lattice sides. So it will be strong and positive at short distances, and it will be, let's say, 0 at large distances. So the particles at low temperature will have a tendency to agglomerate. They will cluster. And if I look at the profile of density as a function of position of a space, this is the 1D case, but you find the same thing in 2D. You will see a bump of liquids surrounded by very low density vapor. And if you go at high temperature, then you will have, through a first order phase transition, a uniform profile of density of a space. Okay? So it's like having a Ising model at fixed magnetization. So this is the case of a single environment. Now the question, what happens when you have many of them? So what you do is that for each new environment, you just randomly reshuffle the positions xi and xj. So for the same two neurons, they will have position there in the first one, and there in the second one, and so on. And then you add all these matrices together, and you ask what happens. So you get a phase diagram, which is similar to the one of the Hopfield model. So here we have alpha. Usually in the Hopfield model, alpha is the number of fixed points divided by the number of neurons. Here, this is the number of environments divided by the number of neurons, and here you have temperature. And at low enough temperature and low enough load ratio, alpha, you are in a phase where you can actually have a bump of activity in one of the environment. That means that the neurons which are active have positions or have place fields which are close to each other in this particular environment. Okay? So they code as a population in a coherent way for one particular position. And if you look at the same neurons, the position of the same neurons which are active in another environment, they are just all scattered all over the place. That means they don't code for any position. So you can see that, for instance, in this simulation. You can yes. So this is a simulation with 3 environments, which was done by Sophie some time ago. So here what you see is that you have a certain number of neurons, 8,000 neurons. And what you see is that so we have a single Monte Carlo simulation, and we just represent at any time the neurons which are active, and we put a dot of the corresponding color. So there are 3 colors because there are 3 different environments. And when they are on, when they are 1, we put a dot at the position of the center of the place field of this particular neurons. So this is you see 3 times the same configuration of neurons, but just represented in 3 different ways. And you see that then you have a bump of activity, so it's coherent in the blue environment. Sometimes it jumps to the red and so on. So you see clearly it's possible to have a continuous attractor. The bump will diffuse. And sometimes it jumps spontaneously to the other attractors. Okay, so it's fine. We have the point 1. We have the memory on the attractors, but we don't have the driving force from one attractor to another one.
Is the form of this function J?
J? Oh, as a function of a distance? So in the model I showed, that was simply something going from, let's say, 1 over n to 0. But you can use anything which is short range. It will give more or less the same thing. The important thing is that you have short range attraction and long range inhibition. So you maintain the global activity of a network, and you will get a bump. Okay, so what about now the inputs? Okay, because in the experiments, we have different attractors. We have 2 of them. And then we see jumps from one attractor to another one, but they are triggered by the light switch. It's not just spontaneous. Okay, so how can we understand that? So the question is what are the kind of different inputs which are incoming into the hippocampal network? So there are many different inputs. So for instance, when we move, we have visual inputs. So we see the environment around us, and we use these visual inputs. But of course, even in the dark, we can know more or less where we are through a mechanism which is called path integration, which is essentially an integration of our motion, and it gives us some approximation of where we are. Usually, if everything is okay, the 2 inputs are coherent. They don't get into conflict, right? It turns out that in the experiment I showed, we see exactly a situation of conflict. And people like that because they can understand what is the relative importance of each kind of input. So one way to do that is an experiment here, which is now very popular. So people use virtual reality in order to actually create some mismatches between the 2 sources of inputs. So you put a rat on a ball. The ball is actually rolling. And you can show a movie to the rat, and the movie, I mean, the way you present the movie should be in agreement with the distance which has been run by the rat, okay? But of course, in virtual reality, you can disaccouple the two speeds. So for instance, you can have the movie running twice faster than the position of a rat, or twice more slowly, and so on. And you see what is going on. And if you do that, you see actually what is going on for place cells depends on the place cells. So some place cells are clearly place fields. They have place fields in the referential, which is attached to the movie. So this is an example here. So what you see here is a place field in the coordinate of the movie. So when you have a movie which has a speed which is 1/2 the normal speed, you see that the place field has not changed, it has not moved. But another place cell actually has changed position. So the place field was clearly driven by the path integration input. Okay? So it seems that all these different sources of inputs go onto the place cells, and they go with different strengths. That means that some place cells will be very sensitive to visual inputs, other one to the path integrator, and so on. Of course, there are other sources of stimuli. There could be olfactory stimuli, many other things. Okay, so the path integrator, it's a very complicated story. It's not very clear where this path integrator is. Some suggestion is that it's in some cortical area, which is where grid cells are, but this is not very clear. This is an area which is very close to the hippocampus, which is projecting information to the hippocampus, and which is also getting back projection from the hippocampus. But anyway, so in the model we do, it's some kind of ridiculous model of this, where we essentially model this through some fields. So remember, we need the couplings in order to have these continuous attractors, and the possibility of having different continuous attractors in the same network. And now we use the fields in order to drive the activity along these attractors. So we have two contributions to the field. One is coming from the external input, the visual input, and there are two kinds of visual condition, A or B. So whether it's A or B, the visual inputs will actually activate some fields incoming onto the neurons which code for this the position of a rat X at that time in environment A or B. And we have also another source of inputs which comes from the path integrator. And both usually are coherent, but they might be in conflict. So they are coherent before the light switch, they are coherent long time after the last switch, and they can be in conflict. Okay? So you can imagine that you have this network which encodes two different attractors, and now you are projecting inputs which wants, when you are in the conflicting phase here, which wants to activate the neurons which code for position X of a rat in environment B, and this is exactly what the visual input is doing, because the light condition after the switch is on B. And the path integrator is still in the old environmental representation, so it's activating the neurons which code for X, position X, in environment A. And they are in conflict. And if the two strengths of the inputs are more or less the same, then you will see some jumps from one to the other. I can show you a simulation. And then I will stop after that. So when you see this, so here you see it's always the same kind of plot, a simulation. So the red cross is the position of a rat, that's the experimental position, just to make this realistic. And then we have a model with 400 neurons. And we have two sources of inputs, exactly as I described. And you see the jumps from one environment to the other one, and which is always compatible with the real position of the rat. Okay, so this model makes two predictions, which we could compare with the experimental data, and then I will stop. I will show you. So the first prediction is somewhat obvious, but it was not in the experimental paper. So what was in the experimental paper reported is exactly this curve. So what people looked at is, they looked at the probability. So suppose you are in A, light A, you switch to B, and you look at the delay tau after the light switch, and you ask what is the probability to be in the same environment as the one before the light switch. That is not the correct one, okay? And what you see, you take all the data, and you do the analysis, and you plot this, and you get this kind of curve, which is some kind of exponentially decreasing function, more or less, which decays over 5 seconds. Okay, so it seems that the longer you wait after the light switch, then the less likely you are to be in the previous environment. But in fact, this is a convolution of two different phenomena in the model. It seems to agree with the data. One phenomenon is the fact that there is a mismatch between the visual and the passive integrator input. And that lasts for some exponential distributed time. And when you are in this mismatch, in this conflicting situation, the frequency of flickering between A and B is just constant. It's not decaying over time. It's like a two-state model. You have two states, and you can jump from one to the other. And as long as the two states are there, then the frequency of flickering is always the same. And this is clearly what we see here. It can last for 10 seconds. We see something which is constant in the data. So it's much more similar to a two-state model than something which is decreasing like that. And the other consequence in the data, which I find interesting, is this one. If we look at the situation before the light switch, then the path integrator, the internal and the external inputs are coherent. So everything is fine. We have a single nice map. And if we look at the error in position decoding, it's rather small. So here 14 centimeters is not too small in a 60 centimeter wide box, simply because we don't have many neurons. But let me call this small, okay? If I look now at the time right after the light switch, I told you that, in fact, if we decode with the place fields corresponding to the map, which is activated at that time, the error is small. That is true, it's small, but still it's a little bit larger. It goes from 14 something to 16. So we have still a small increase in error. So how do you get a small increase in error? You could say, well, everything is more noisy, so performance are worse. But actually, this worsening of performance is not due to noise. It comes from another conflicting signal. Because what we can do exactly the opposite of what we should do. We can decode the position with the map, which is not the decoded one, so the wrong map. Obviously, if we do that in the coherent phase, that's terrible. The error now is about 34 centimeters, which is half the size of a box. But if we do that during the conflicting phase, it drops down from 34 to 29. Still, it's really bad. But it's not as bad as before, which means that what is creating the error here, the increase in error here, is actually a signal corresponding to the wrong map. If you use it and use the wrong map and decode the position of that, you do a little bit better than in the coherent phase when you don't have any input corresponding to the other map. So I'm not sure I'm clear. So what I'm saying here is that you see an error here, but this error is signaled in the wrong environment. And there is very clear evidence of that. I think I should more or less stop.
You have some time.
Okay. But I don't take it. Okay, anyway, so I think what this work provides is some indirect evidence for attractor neural network theory, or let's say with multiple continuous attractors. This is not a proof, obviously. But I think there are more and more evidences like that now in the literature. So it seems that it's very old ideas coming from the 70s and 80s now are more and more supported by data, which I think is very nice. But of course, there are many things which are missing. So first of all, what would be very nice is to have experimentally a joint recording in the hippocampus and also in the path integrator region. Then it would be much stronger to see whether this theory is correct or is wrong. And from a theoretical point of view, I think a lot of things are missing. Because here, you see, what we do here, we assume some specific function of a distance, and we build a synaptic network with this prescription, which is very similar to what John Hopfield did in his famous paper. But we know that doing that is actually not optimal at all. When you do that, you have a capacity which is 15 times smaller than the optimal capacity. So it's clear that doing this is not good. So far, nobody has tried to really understand what is optimal. And this is not only a purely academic question. I think it's an important question because there are many issues which are not understood. So a continuous attractor, so sometimes it's true that we really want to memorize something which is more or less continuous, let's say homogeneous in space. But there are clear evidences also that when a rat is moving in an environment which has, let's say, attractive spots, like places where there is food, water, and so on, then there are many more place fields which are around these interesting spots than in regions which are, let's say, deprived of any interesting stuff. And we probably do the same thing. We have a very precise maps of interesting regions, and we don't care about, deserts or whatever. So that means that having this idea of purely homogeneous, uniform continuous attractor is not a very good representation of reality. We should have something where we have non-homogeneous attractors with interesting spots. So we have to remove the spots which come from the interference with the other maps, which are due to this rule here, which is far from being optimal. And we have to add spots which come really from interesting functional regions where the rats want to go. And this is something that is not existing in the theory. And I think there is plenty of things to do more in future. Okay, so we stop here.
Thank you very much. There's a question in the last row already, so you have to climb a bit.
Hello. Look, I have a question. On the very, like at the very beginning, you showed the matrix of the spikes, experimental spikes taken from the brain of the mice, right? First, I would like to ask you if you know Demian Battaglia.
Yes.
Okay, well, because I already seen this kind of experiment in some of his papers. If you can go to the, because I'm, this one. I'm interested in this kind of representation in a more general context, and I would like a little bit, I would like to ask you if you tried to compute the overlap matrix, means scalar product of the rows. No, sorry, of the column.
The scalar product of a column?
Yes, one column and another, no? And you do the overlap matrix.
On the next one? Yeah, so they will be correlated, obviously, because the rat
but is... can I see?
No, I don't have it here.
In fact, I didn't see. Because they did, okay, maybe after, it's a little bit long to explain. Let's say I study this kind of representation in the context of replica symmetry breaking, okay? And the nice thing that Demian obtained is that this thing evidentiate some kind of structure of the overlap matrix. And I was very surprised when he spoke here some months ago, because I derived a similar kind of structure in the context of my research, which is totally theoretical, to represent what is called finite volume pure states. So what are the, I tried to use this representation to talk about the pure state of replica symmetry breaking in finite volume. And the interesting thing is that this kind of experiment, if you do the overlap matrix, some kind of replica symmetry breaking is observed. Did you try it?
No, I haven't tried that.
Okay.
I haven't tried so complicated things.
No, it's just, sorry, it's just...
Yeah, maybe
you can talk about it.
Yeah, you can talk about the project later on. There's another question. Micro, please.
The dynamics, if I understand correctly, is relaxational, no? I mean, the couplings are symmetric at the end. So when you have a manifold that is like this, a bit flat, if you added something that is not, wouldn't it take you for a ride on this manifold?
Okay, so it depends very much of the dimension. So in 1D, so the place fields are known to be directional. So it means that if you have a spot on the corridor, if a rat enters, for instance, this way, then the cell will be active. If it enters this way, it won't be active. So then you can imagine that when you have a rat running on that, you will have highly directional interaction. In 2D, it's much less true, in the sense that it seems that since the rat can enter the same zone in many different orientation, then it's not directional. But there is information about the direction in other cells, which are called head direction cells. So they could drive the network, even if you have some kind of symmetric couplings in one direction, along one direction and the other. So this is something we are not considered, but this is, there are extra information which is not here, I agree. Yeah.
Okay, next question.
Experimental question. Is there redundancy in the place cells, which means a certain place, is it mapped in several cells?
Oh, yes.
Yes.
Oh, yes. We don't see it too much. We see it because otherwise there wouldn't be correlation, right? But we don't see too much because we have only 30 cells in the experiments. But it's plenty of them, yes.
Yes. And the other question is there, I mean, what is the memory? For example, that a place, say if you repeat the experiment, I don't know, 1 day later or something like that, does the same cell remember the same place, or
Yes.
is it a
Yes.
program? Yes.
This is not true for all rats. It's a little bit fuzzy. I mean, there is a couple of papers where people look at the influence of the age of rat. So you can see that it's basically drifting away. But for young rats, then it's quite stable. Yes.
But there is no reprogrammation going on.
What do you mean by reprogrammation?
I mean, very late times, the same cell records for a different place.
I don't know. I mean, people have experiments which last for weeks, and I don't know what will happen for years. I mean, that's it's quite possible. Maybe I mean the network fades away. I don't know. It's quite possible. There will be some palimpsests.
Maybe I have a small question. Remi, what about if one could do the experiment on humans? Would one get the same kind of results?
So there are some experiments on humans. I mean, in people having epilepsy. I mean, to very high serious degrees where they are able to do some ablation and so on. So if the people are voluntary, then some recording is done with electrodes before the surgical operation. And so place cells exist, that is clear. But obviously people don't have, multi-electrode recordings, so they can't not measure networks or things like that. But it's quite likely, I mean, there are the same things in monkeys and bats and, So, I mean, there are some variations, but the place cell exists probably in all mammals. Even there are some evidence for that they exist in fish. So, I guess it's exactly the same thing in humans, for instance.
Okay, so are there more questions? So I think not. So let's thank the speaker and all speakers of the first day.